Good morning. I'm opening this session on AI, and I thought it would be useful to get all converging on two or three concepts that I think will be usable for the rest of the presentations. One is make sure that we all understand that within the field, let's say, of statistics, mathematics, all these terms are related, and they are the children of the AI concepts from the 60s, 50s. And deep learning itself is just a subdomain of machine learning. And what has changed here is two things. One is the amount of data, and the other is the capacity of computing. But if we go to some of the classics, this is just statistics, mathematics at a scale. The other very important concept, because I will use two case scenarios, is machine learning versus deep learning. And the difference is right there in what we call feature extraction, that is a heavy burden process that includes a lot of intellectual requirements, domain experts, which is basically formatting the data in a way that can be processed later on by the computers. The dream, the capacity of deep learning is just, and that's a just, be able to remove that burden of the human trying to create the features. And this is achieved, again, by massive amount of data that allows to avoid this feature extraction, this engineering of the data for the purpose of the analysis. So basically, it frees the user from thinking too much. Now, the thinking is still at the beginning, and the thinking will still be at the end, and maybe much more thinking and processing of the data entry, acquisition, and interpretation than we ever expected. Now, the other important message, because I get asked this every week, is that these machine learning, deep learning models, they can explain, and explain in the terms of mathematics. So they can create a model, they can classify, and they can predict. They cannot establish causality. That's mostly today left for biology, for the wet lab. So when somebody comes and says, I have this data, can you tell me the mechanism of action of this drug? The answer is no. I can tell you how this looks like, but I cannot tell you the mechanism of action or the causality of this disease so easily. So remember that, again, this is a classifier predicting type of analytics. So where do we start for genomics? We have 20 years of accumulation of increasingly complex and rich data sets, whether genomic, transcriptomic, epigenetics, etc. And much of this data can be seen as extremely well formatted for analytics compared to, for example, say, medical records. So this is a great advantage for the field, for AI in general, for genomics. And from this type of data, we have, again, what is an ideal situation for deep learning, is that deep learning can get away with just simple feeding sequence. So basically, what we call the training, testing, validation schema will just say, give me just plain sequence. There's no feature engineering necessarily, so we just can feed the feature. Then there's a number of, now tens of structures to analyze this data, and basically then we do very basic prediction analytics, and we interpret. So again, because the data is so well formatted, ACTG, because the data can be in large volumes, even one single genome is already a powerful tool to feed to a deep learning model, one genome, we can actually get away with limitations that are common to other fields in medicine. And in this same primer that we published recently, we already reported that despite of the common understanding, actually there's already been deep learning used at almost all levels of analytics for genomic data, transcriptomic data, etc. Much, much less on medical interpretation, medical implementation of this type of algorithms, but pretty advanced for the basic understanding of genomic biology. So I wanted to document this with two user cases, one maximizing the concept of machine learning and feature engineering. The other one is pure use of sequence and how to transfer it through the space of knowledge of a species. So again, the first case will be on the top, and I will show you the problem of feature extraction and how to go through this challenge. So we completed recently an analytic that does machine learning, where the feature extraction is all the amount of data interpreted by us, by others, scores, every single thing, every single data source that we obtain from the human genome across the time. And we were able to put it into a single model. So every single thing that you see in that histogram is different sources, different data sources that were featured, engineered by somebody else or by us. The colors are either blue being historical scores on the genome, already featured and engineered by somebody else, and the green, red, and yellow are the impact of bringing novel sources of data and how this will change prediction. And prediction here is pathogenic variants in the regulatory genome. So the first thing that we notice in this area under the curve is that everything counts. So whether the classic scores, whether putting all the new information here in orange and the ensemble processing of those things, which teaches us one thing, is that as we get more and more data and more diversity, we keep adding. We keep adding detail, we keep adding accuracy. And everything, to some extent, contributes, as you can see here. These are the relative contributions of any of one of those features. And then we challenge ourselves to what is probably the most important thing, is how robust, how can we generalize these findings? And there is the training, there is the testing. You can see it performs well. And then we try to generalize to distant or unrelated sources of queries, in this case, new pathogenic variant sets, or even pathogenic sets in completely different systems like non-coding RNAs. And you can see that always in gray, we can survive some of this generalization with more or less success. So overall, you've seen here the features of machine learning means data, many types of data, everybody contributes, everybody adds to accuracy, and then the pain of trying to generalize and convince ourselves that this actually performs in our data or in the data from somebody else. Why do we do this? Because then we can start moving into the clinics and say, okay, now we created this global score by learning from so many features, and we can start positioning new sets of mutations. These are Mendelian variants, and you see that they come towards 100. That's the most highly predictive for deleteriousness, and it works. And we think these are the type of things that will move to the clinics for the non-coding genome. And also we challenge ourselves not only to see how does it perform for what was trained for, that was deleterious variants, but how does it perform more generally on function. And what we do with BeamRAN at the university is large screens of enhancers, something that is very difficult, very fishy to study, and we can quantify the enhancing capacity of 50,000 enhancers in the genome. And then what we do is combine the prediction of functionality essentiality, so the most important parts with the most regulatory active enhancers, and we start getting this type of cross images where the most essential parts of the genome that are regulating something actually are in front of genes that are Mendelian disease genes. So here, the prediction will be that people presented with a clear presentation of a Mendelian disorder for which we find no coding region variant, the next place to go and look for will be the corresponding enhancer. Why? Because we know it's functional, and second, we think is essential. So that's the first user case to try to typify the concept of feature learning and feature acquisition of any type of data, whether it's about chemical, structural, genetic population. And the second one is more proper to the magic of deep learning. That means give me just the raw basics and we'll take care of it and everything's going to go just well. And that case is a case that I like very much. It's called transfer learning, meaning learn in the model that you know the best, in this case humans, and then just walk along the species and try to propagate the human knowledge into other species, including as far as you could imagine theoretically. Here is on the bottom, that means we just use raw sequence, and you will see soon some surprises. And this is the way it looks. We, for example, here I'm representing promoters, which is actually very easy because they are so characteristic in their sequence use. And we train test a model with an accuracy of 83%. That means that just on plain sequence, the computer knows that this is a promoter, this is not a promoter, at 85% accuracy. And then we do the first jump in the transfer model. It says, can I discover the mouse promoter? And all I do is put the mouse genome through, and it will call, and we dropped 10 points of accuracy. So that means that the mice promoter, without knowing anything about the mouse, in case we know much about the mouse, is the only other genome that we know extremely well, we can already predict with an accuracy of 7%. We have now more or less in working order to go through enhancers, exons, chromatin, open chromatin, same concept. And what we are testing is how far can we go along the mammalian line and still get appropriate recognition. Now, why do we do this? Just because of what I mentioned is that there are only two well-annotated genomes. That's humans and mice for the whole genome. You have excellent prediction for exomic, for protein coding, but not for whole genomes across the species, even for things like zebrafish or C. elegans that are heavily used, and even less for agriculture. So transfer learning. Now, those were two cases just to typify machine learning, deep learning approaches and how they may work and how do they work. But I wanted to highlight two notable progresses that tells you a bit more of where these things are going and how they are going to fit. And the first one is a large concept of new biology. And this is a paper from about 1 month ago from Illumina, from their group of artificial intelligence. And it's very impressive because what they do is just they learn by deep learning from transcripts, and they figure out the computer learns to identify splice forms and cryptic sites, and then they correlate that with pathogenicity, and what they find is that they can predict pathogenic mutations. Many of them are not known. Actually, they estimate that 10% of pathogenic mutations in patients with some of the rare disorders are actually cryptic sites that the computer can distinguish. Now, why did I say this is new biology? This does not sound like new biology. We knew about cryptic sites. Well, the new biology aspect is that even though they were just injecting sequencing into their programs, what they did was change the window. So they started with what we know, things that happen around the script, the splice sites. And then they start getting broader windows. And they were very surprised that actually very large windows of sequence, so injecting 10,000 base pairs of sequence actually had the best prediction, meaning that there's a lot of biology that is unregistered in our mind, in our textbooks. And this is what deep learning can do, is that, like we heard with the retina images and female-male, is that we see things that we did not recognize. And I think this paper was a very good example of learning about a splice variant determinants beyond and far away from where we look for. And the second notable case is from Raquel Diaz and Ali Torkamani, my colleagues, where they we're solving something that is commonplace, imputation, done by UK Biobank, done by anybody that is running on very large data sets of genetic genotyping data. And they gave me 2 slides, one that shows that equally accurate, doing by deep learning or by classic imputation statistics. And that slide, I did not show it because I can tell you it works the same. What I wanted to show you is that it works faster. Why? Because we thought that deep learning was just a huge footprint on computing resources. And actually what you can see is a bit of the reverse of what we thought, is that actually you can run things rapidly and efficiently at the same accuracy. I think this is very important data. Now, we have taken care at the Scripps for the translational part, and we became acutely aware that much of this new data and scores, etc., are very difficult to retrieve. So we created this browser where you can pull out many of these scores, and the group at Illumina just placed their new scores called Primate AI and Splice AI into the same browser. So you can actually find at any genetic position all these new prediction scores plus other useful information and a lot of NLP-based natural processing language readout of information. So I hope that you can use it. So I want to close here with 4 statements. The first one was actually a statement from Nature Genetics editorial that came out with a primer, where Nature Genetics was already predicting that their journal, as many other journals, will be taking a heavy load of submissions using deep learning. So they expect those papers to be flooding the journal in the next couple of years. As it was discussed today, and I mentioned, we have a lot of applications for deep learning in genomics, but not for much for clinical use, and this is something that we're trying to push, like genome interpretation, and it will require proper validation in the clinics. There's a lot of bias, and this is not unique to genomics. And again, the comment that if we thought deep learning was going to take away the pain of thinking, we'll probably have to be thinking even harder because of the inherent noise and danger on these predictions. And as I mentioned, whether you validate in the clinics or you validate functionally, the key of the game is you have to validate. So I would hope that everything that we do with deep learning will rapidly be matched with clinical trials, as we heard this morning, but also with proof from the laboratory that what we predict is actually true, like a reporter and analysis of the predictions. So I will close here, and the work I showed is from these collaborators, from Scripps, from Stanford, and many thanks to Kyle Farr from Illumina, from depositing all his predictors in Omni. Thank you very much.